Analytical methods, okay, are destructive. Analytical methods tell us something either about maybe the structure or the composition of these proteins, and this destroys the protein, right? So if we think about an analytical method, we want to determine, let's say, what is the composition of proteins or what's the population of proteins in a glass of milk. And we can isolate, some milk, separate out all the proteins, so get rid of the fatty acids and get rid of the water and get rid of all the other junk and just be left with proteins. We can use an analytical method, like we'll see, mass spectroscopy, to actually determine the identity of what's in milk. So we have high peaks here of casein. Obviously, milk is high in casein, lactoglobulin, a specific type of antibody, lactoalbumin, which is important because this is what helps to solubilize those fatty acids in solution, right? So all of these proteins that we would expect to find, we do find using this analytical method. Preparative method, so let's start with that. For a preparative method, okay, first what we need to develop is something called an assay. An assay essentially is just a test, right? It's a method to test, is our protein there, okay, and how much of that protein is there. So an assay will vary from protein to protein. So for example, if we take this example here, lactate dehydrogenase, we're going to see this in chapter 16, but lactate dehydrogenase, what it does is it reduces lactate to pyruvate, or rather reduces NAD plus to NADH, and oxidizes NAD to pyruvate. Right? So lactate is oxidized, NAD plus is reduced to NADH. It's a redox reaction. And luckily for us, okay, NADH has a characteristic absorbance. Okay, meaning that it absorbs light at a specific wavelength. So by measuring, shining a light through our test tube here, that light at that specific wavelength, we can measure how much of that light isn't transmitted to the other side of the test tube. That's how much was absorbed. Right? In this case, that corresponds to the amount of NADH that we had. So in this example, we can measure the progress of the reaction. So say that we take lactate and NAD plus in solution, we add some lactate dehydrogenase, we can measure the progress of the reaction by measuring how much NADH is formed per time. Okay, so this would be an assay to say, okay, well, we have lactate dehydrogenase because we're measuring an increase in NADH. If we didn't have it, we wouldn't measure an increase in NADH. Additionally, the rate at which the levels of NADH rise tells us something about how much lactate dehydrogenase is active at that time. The more lactate dehydrogenase we have, the more NADH we'll make in a given period of time. Right? So this is one example where we can use something very convenient to measure the activity of our enzyme. Now, it's not always going to be this, obviously, because not all enzymes use NADH, but we have to have some sort of method in order, or some sort of assay, rather, to measure the activity of our enzyme. Right? This is going to help us to follow, as we'll see when we talk about quantifying protein purification, or preparative methods. Right? This is going to help us monitor how our purification is going. Right? So an assay is just a test. Now, to purify protein, we need a method of separating that protein, or proteins from one another, right? The first step in this process is typically something called fractionation, okay? So where are our proteins going to come from? Well, unless we're, synthesizing them in bacteria using recombinant DNA technology, usually the protein that we're going to be getting is coming from a cell, right? Let's say that it's coming from a eukaryotic cell. Well, what do we know about eukaryotic cells? Eukaryotic cells have, obviously, organelles, right? Organelles are different from each other, otherwise we wouldn't have any organelles. So a population of proteins in each individual organelle, or the cytosol, varies, okay? So the first thing that we want to do is to purify out the specific cellular compartment that our protein will be found in, or we might not know which it's found in. Either way, fractionation is going to allow us to do that. So what do we do first? So here, our first step is to form a homogeneous. Right? And what is a homogeneous? Essentially here what we're doing is we're using either something called sonication, in fact it's almost always sonication, or mechanical homogenization, where we use a mortar and pestle to grind the cells. And here all we're doing is we're breaking down the outer phospholipid bilayer, right? So we're breaking down the cell membrane. And we're forming a homogeneous mixture that's made up of everything that was inside the cell. Okay, so we have the organelles, which remain intact. We have the cytosol, which is now just part of the solution. And of course, we have some remnants of the cell bilayer, the cell membrane. Okay, but now we have a homogeneous mixture that contains all of our organelles in solution. And this is important for the next step. The next step is to take this homogeneous into centrifusion. And we're going to centrifuge it in increasingly higher centrifugal fields. So we'll start at a centrifugal field of, say, 500 times gravity, 500 G, right? And what's going to happen now is that the densest organelles will begin to pellet, right? So the density of a given organelle determines how it will sediment at the bottom. So here, spinning it at low speed, right? I know 500 G seems high, but in the context of fractionation, it's quite low. So now only the densest material will be forced to the bottom of our test tube, and everything else will remain in solution. So it happens that the densest organelle, most of the time, is the nucleus. So at low centrifugal fields, at low speeds, the first thing that we get to pellet is the nuclear fraction, right? The reason that we call these fractions is because it's a fraction of what we started with. Okay, we started with a full homogeneous, now we're collecting a fraction of that, specifically the nuclear fraction. We're going to see fractions later on when we also talk about chromatography. But we can collect this pellet now and keep that in mind and say, okay, all that we have here is nuclei, and all that we have left over in the supernatant is everything that isn't nuclei, right? So we take that back, we put it into our ultracentrifuge again, we'll spin it at higher G, so 10,000 G, let's say. Now what we get is the next densest organelle, which in this case might be the mitochondria. So that will settle. Everything less dense than the mitochondria remains again in the supernatant. We can collect the mitochondrial fraction, keep that aside. Now we have two. We have the nuclear fraction, got all of our nuclei. It's got our mitochondrial fraction, which has got all our mitochondria. Now we have everything that isn't either of those left over. So we can do the process again. So we can spin it again for, say, 100,000 G at 1 hour. And now we can collect all the microsomes, right? So the microsomes will come down. Now we have 3 fractions. So now we have a bunch of fractions, right? We do this over and over and over until we have a bunch of fractions, each of which is comprised mostly, right, not 100%, but mostly of nuclei, mostly of mitochondria, mostly of microsomes, mostly of ER, and so on and so on and so on, right? So we call this subcellular fractionization, right? And the method here is called differential centrifugation, so that we can collect progressively denser components of the cell. Right? And now we can analyze each of those. So we can take our nuclear fraction, mitochondrial fraction, microsomal fraction, we can analyze those separately and say, is our protein here? Right? Say we're trying to study protein X. So we can look now at each of these fractions and say, do we see protein X? If we do, then we know in the nuclear fraction, we say, okay, this is a nuclear protein. Or in the mitochondria, right? What might we say we see protein X in the mitochondria? What is it likely involved in? What happens in the mitochondria? It's a meme. The powerhouse of the cell, right? What does that mean? What goes on in the mitochondria? Metabolism, probably some kind of metabolic enzyme, right? So this gives us a starting point, right, to start analyzing our protein. Where do we find it, and what is that organelle responsible for? Now, once we have this, okay, well, each organelle obviously doesn't contain only one type of protein. It contains from dozens up to thousands of different proteins. So how can we now separate our specific protein away from the rest of the proteins that we are not interested in studying right now? And this involves a method called chromatography. Now, chromatography is an umbrella term. There are several different types of chromatography that we're going to talk about here, right? And the first is size exclusion or gel filtration chromatography. They mean the same thing. Right? And in size exclusion chromatography, as the name implies, we're going to filter out proteins based on their size. Not on their length, but on their size. How big are they? And how does this work, right? So all chromatography is based on the same general, apparatus here. We have our chromatography column, right, which is just a glass or a plastic tube, right, usually about, well, depends on how much you're doing, but it could be either a millimeter in diameter up to a centimeter in diameter, right? So it can be smaller, it can be large, depending on what how much we're trying to purify. Now, inside this glass or plastic tube, we put what's called the resin, okay? The resin is what determines what kind of chromatography we're doing. In the case of gel filtration chromatography, the resin is made up of something called, like a brand name, Cephidex, right? It might be a different name, but it's like Pampers, right? It's called Cephidex. And Cephidex here, okay, has a very peculiar structure. Okay, so here we're seeing a very zoomed in view of what's going on. We have these tiny little beads, which are the resin, and they're about the size of a grain of sand, right? Or smaller, depending. Okay, so they're very small, and we pack them into this glass column here. Each of these beads in gel filtration chromatography isn't solid. Instead, if we take a cross section, what we see is it's got many channels going through it, right? And they're not as simple as just going, you know, from top to bottom. They actually look like this, right? So it's kind of like a maze, right? The way that they're manufactured makes them porous, but they always have an exit and an entrance, right? So what's going to happen? Small proteins, once we feed them over the top of the resin, right? So here's our protein mixture that we isolated from maybe our nuclear fraction, let's say. What's going to happen? We'll drip that down onto our resin, right? So here's our protein sample now, and it'll filter through just by gravity. It'll fall down through the resin, right? Now, small proteins can enter these beads, right? And now they have to make their way all the way through these channels in the beads before they exit, okay? Then they'll find another bead and they have to make their way through, then they'll find another bead and have to make their way through. Larger proteins cannot fit into the pores of the Cephidex beads. So they get to flow right around, right? They flow around, they find another bead they can't fit, so they flow around and all the way through. So effectively what happens here is that a small protein has a larger or a longer path from top to bottom because it's got to make its way through all those channels of the beads, while larger proteins can go right around them. So at the bottom here, right, which ones are going to come out faster? Right, the larger ones move faster through the column. So we allow our protein sample to flow through here, right, through the column, and we can collect different fractions. Now, at this point, we're just going to collect fractions based on time, right? So we'll collect whatever flows through for 1 minute, and that's our first fraction. We'll collect for another minute, that's our second fraction. We'll collect for a third minute, and that's our third fraction. The first fraction will be the biggest proteins. The second fraction will be smaller proteins. Third fraction is smaller yet, and fourth is smaller even than that, and so on and so on. So we have now a collection of fractions, each of which is different based on size, right? So here we have size exclusion chromatography. So say that what we're trying to collect here, right, is our green protein, right? So we would collect, say, for 1 minute here. Okay, we move these aside, collect for another minute, maybe that's where our green protein is going to be, and then we collect for another minute, and that's where our pink protein is going to be. But we only care about the green protein here in the middle. How do we know in which fraction our green protein will be? How can we figure out, right? So we have this collection of, say, 10 fractions. We've collected them over the course of 10 minutes, so each fraction is a 1 minute collection. How do we know which fraction is going to have the protein that we're looking for?
By the density of the
Huh?
maybe by the density of the sample of the fraction.
The density, well, it depends on many different factors, right? So if we have a lot of volume up here, then the pressure will be higher, right? So it will push down faster, right? Of course, as the liquid level starts to go down, the pressure decreases because now there's less weight on top of it, right? So we can't rely simply on density or time, right? So we have all these fractions. We know that our protein must be there somewhere. How do we tell where our protein is? Right, exactly. This is why we need an assay. We can take all of our fractions, we'll perform an assay on them and say, all right, well, in fraction 4, our assay indicated that our protein is there. So now there's our assay. Now, here, we're not quite done yet. Okay, so yeah, all right, so let's say that in fraction 4, we see our assay indicate that we have our protein, but gel filtration chromatography doesn't have high enough resolution. In fact, you don't want it to have high enough resolution because then you have to have, you know, potentially thousands of fractions, okay? You're gonna get a range of proteins, right, in a range of sizes, right? So say that our first fraction is, you know, 200 kilodaltons to 220 kilodaltons. Second fraction is 180 kilodaltons to 200 kilodaltons, and so on and so on. So there are many proteins that have those sizes. So now in this fraction 4, okay, yeah, we do have our protein, but we also have other proteins, right? We might have hundreds or dozens of other proteins. So what happens is that different chromatographic techniques or different purification techniques are stacked on top of each other. So here we purified it decently, right? So we have gotten rid of a lot, we've gotten rid of proteins that are way bigger and way smaller than our protein. But we can go further. Let's say we need to purify this even more, well, we can do another type of chromatography called ion exchange chromatography. And this is very easy to visualize, but a little bit more difficult to not explain. Well, let's just go through it. Okay, so let's say that our protein X, right? Our protein X, we look at the sequence of our protein X, and we see that it's positively charged, right? There's a high concentration of, say, lysines and things like this, right? So at pH 7.4, or whatever pH we're doing this at, we see, okay, well, our protein should be positively charged at this pH, right? So we've gone through how to do that. Ion exchange chromatography allows us to leverage that, right? So if we know that our protein is positively charged, what if the resin instead wasn't porous like it was in gel filtration chromatography? What if instead we just add a charge onto our resin? Okay, we make it solid so nothing gets lost inside of there, and we just make it negative in this case by adding a carboxy methyl idea, right? So here our resin in this case will be made of something called cellulose, which we'll see in chapter 11, or agarose, right? If you ever run an agarose gel with DNA, it's the same thing, just made into a very tiny bead, and we'll covalently link a carboxy methyl, giving it a negative charge. So now, we'll take our fraction 4 from our last step, the gel filtration, and that will be what we run through our ion exchange. So now from those, dozens to hundreds of proteins that were in fraction 4, only positively charged residues, or positively charged proteins rather, will be attracted to the negatively charged resin, and they will stick. They will not migrate through the column, right? But other things will. Negatively charged proteins obviously can't stick, right? Negative and negative repel, or neutral proteins also won't be attracted, so they will flow through. And that's what we call our very first fraction, you could say, is our flow through. Everything that doesn't stick in this case will be our flow through, because it just flows right through the column. Everything else sticks. But this is a problem because the whole point here is that we want to, what? We want to isolate our positively charged protein. Okay, so how do we do that? Right? What can we do now to this state? So let's imagine all the negatively charged proteins, the pink ones, are gone, all the neutral ones are gone, so all we're left with is basically this. Okay? What do we do here? How do we get the positively charged proteins off of the negatively charged resin? Wash it with salt. Why? Right. So in chapter 1 we saw salt is able to neutralize the repulsion between the phosphates in the DNA backbone by neutralizing the charge, and the same exact thing happens here. Now, you might be asking yourself, well, if we're interested in having the positively charged proteins, why wouldn't we have a positively charged resin so that we can just collect the flow through and then we have our positively charged protein, right?
Because neutral would also flow through.
Neutral would also flow through. And let's assume that neutral we can ignore, right? But that is true, right? We'd be getting all of our negatively charged proteins, or positively charged proteins rather, right? We'd be getting all of them, but we don't want all of them, we want ours, right? By doing it this way, now we get rid of all the negatively charged proteins, and we can slowly begin to elute progressively more and more positively charged proteins. And how do we do that? Well, we just add increasing concentrations of salt, right? So we'll wash, so after we've gotten rid of the negatively charged proteins, we can add, say, a 1 millimolar solution of sodium chloride, right? And this will negate the interaction between weakly positively charged proteins, right? So ones that are maybe, have very slight positive charge, 1+ or 2+, something like that, maybe they will be washed away with the 1 millimolar salt, and we can increase it to, say, 50 millimolar NaCl. Well, now we get more positively charged proteins. So now we're looking at maybe 5+ or 6+. We'll watch it again with 70 millimolar. Maybe now we're looking at 8+ and 9+. Right? So you add more and more progressively higher concentrations of salt, and we begin to elute higher and higher charged proteins, or more negatively charged proteins. If the situation were reversed, we can also have a positively charged resin if we're trying to isolate a negative protein. So now we can collect fractions instead of just collecting everything all in one go in the flow through. Right, so now, say that in our ion exchange chromatography, again, now we do our assay to figure out which fraction our protein is, and we say it's in the 6th fraction. So what is the 6th fraction? It's a fraction of the 4th fraction from the step before, the gel filtration. So we have a fraction of a fraction. We've now purified a purified protein. So we should have, instead of hundreds of proteins, maybe now we have 20 or 30, right? Or maybe even less, right? But for the best results, right, even now we want to go on to a 3rd step of chromatography, affinity chromatography. And affinity chromatography can be done in several different ways. Actually, we've done it in a bunch of different ways. But the crux of it here is that affinity chromatography relies not on the charge or on some physical characteristic of the protein size. What it relies on is on the affinity of that protein for its given substrate, right? So let's say that now we've gotten fraction 4 from gel filtration, fraction 6 from ion exchange. We can take fraction 6 from ion exchange and run it through an affinity column. Let's say that the protein that we are studying, right, we know binds glucose, okay? Well, what if now we devise a system where we have in our chromatography column the resin covalently bound to the substrate? Well, now when we flow through, when we add our fraction 6, okay, what's going to happen? Our protein is going to bind to the glucose, which is covalently linked to the resin, and it will stick, right? Everything else will flow through. So now we have, again, all of our protein of interest stuck to the column. In order to get it off, all we have to do is to add free glucose. Right? So remember, binding between a receptor and a ligand, or a protein and its substrate, is not infinite, right? We have a KD, so there is going to be some equilibrium between being bound and being unbound, which means that after some time, of course, this will release. But in this state, it will release the glucose, but it'll just bind another glucose that's bound to the resin, so functionally it's the same. But if we add excess glucose, now when this releases, it has a high chance that it's going to bind a free glucose now in solution, and so won't be bound to the column and will flow through, okay? Now, here is using the example of glucose, but this could be anything, right? This could be whatever the substrate is for that protein. So if our protein was binding to testosterone, right, then we could make a resin with covalently bound testosterone. We could bind that, purify it that way. And it doesn't have to be necessarily a substrate, okay? We can rely also on physical characteristics of what we call epitope tags. Okay? Epitope tags are something that involves recombinant DNA technology, and basically say that we want to purify the protein. And in this case, we've isolated the gene of the protein, and we know a lot more about the protein. What we can do is to engineer some DNA sequence, okay, that we put in front of the gene, such that when it's translated, it produces a sequence in the protein that we can target. Okay, so we call that an epitope. So for example, say that we have our gene for our protein of interest. Okay, we'll start with our gene over here. Okay, this codes for protein X. This is DNA. Right, so here's the gene for protein X. Now, recombinant DNA technology allows us to actually modify this, right? So we can modify something in here, or we can add an extra bit of DNA at the beginning or at the end, okay? We determine what sequence this is, and because we know what sequence this is, we know which amino acids it will be translated into once it's transcribed and translated by the ribosome. So we can pick our DNA sequence that we're going to attach now to our gene X, right? And let's say that this codes for 6 histidines. right? So we're gonna add 18 base pairs that code for 6 times 3 is 18, yeah, 18 base pairs that code for 6 histidines. That means that now, when this is converted into mRNA, and the mRNA is translated into protein, what we'll have is the protein sequence of X, the amino acid sequence, plus 6 histidines at the end. Those 6 histidines, we call that an epitope tag. And why is that useful? Well, an epitope tag typically has some characteristic that makes it easier to purify that protein. In the case of histidine, 6 histidines in a row can be recognized by nickel ions. In fact, they're bound quite tightly by nickel ions. So, this is a type of affinity purification. Now we're relying not on the affinity of the enzyme to its substrate, but instead, we're relying on the affinity of the epitope tag to its binding partner, in this case 6 histidines with nickel, right? So what happens here, okay, we add our protein with a 6 his tag, it binds to the nickel, everything else flows through. In order to elute it, right, this presents a bit of a problem because it's binding the protein itself, so we can't add free substrate. But in this case, the substrate is really histidine, and more specifically, the imidazole ring of histidine. So by adding free imidazole, we're doing the same thing that we did by adding free glucose. The nickel will bind to the imidazole, releasing our protein, which can now flow through, right? And there are many different types of epitope tags that we won't go over in specifics, right? We have a 6 histidine tag, there's a flag tag, there's many different types of epitope tags. Affinity chromatography is also powerful because we don't have to rely on either affinity for the substrate or affinity to an epitope tag. We can actually devise an antibody, right? Devise an antibody that will recognize specific proteins. So say that we want to, again, purify protein X, but we don't have the gene isolated, we can't purify it for whatever reason, or purify the gene for whatever reason, so we can't add those 6 histidine tags. But if there's an antibody available, there are hundreds of thousands of commercially available antibodies. If there's an antibody available for protein X, well, now we can order that antibody, right? Sometimes you can make the antibody, right? So your antibodies, you can make antibodies, not that difficult. That antibody obviously is going to recognize protein X. So we can take that antibody, covalently link it to a resin, right? And now we have a resin with antibody. Right, so here's our resin with our antibody. We flow our fraction through. This is going to bind now to protein X, and because antibodies are specific to a specific antigen, everything else flows through, and now we have the same situation that we have here. Right, now to elute this is actually quite interesting. All we really have to do is decrease the pH, because this is going to get rid of the interactions here between the antibody and the antigen, right? And now we can flow through our protein. So affinity chromatography is quite powerful. There's many different ways to do it. It relies on either, again, specificity to the substrate, specificity to an epitope tag, or specificity or affinity to an antibody. That's a very powerful, very versatile tool. Now you might, if you're working in a lab, have done HPLC or heard of HPLC. So all of these chromatography steps, right, they all rely on how many sites of interaction we have, right? How many sites of interaction do we have? Because what's going on when our sample flows through our chromatography column, right? So here's our chromatography column. Here's our resin. It's a little filter at the bottom so that the resin stays put, but the stuff can flow through, right? Our mobile phase can flow through, okay? How many beads we have determines how many sites of interaction we have. So the finer the bead, right, the more beads we can fit in that volume, and the more sites of interaction we have. But this is going to present a problem because eventually we're going to have these packed so tightly that gravity is no longer able to pull it through, right? It'll just be basically solid. So HPLC, high performance liquid chromatography, essentially just adds a pump, right? And this pump is connected to our, say, reservoir, right? It's not this simple, obviously. And this then forces the liquid through this fine resin. And why do we care about the resin? Because the more resin we have, the more sites of interaction we have, and the more sites of interaction we have, the higher resolution we can get in our fractions, right? So now we can collect fractions in terms of seconds, let's say, right? And here, for example, we have absorbance at 220, so we're just collecting protein without any kind of assay or whatever. Let's ignore the type of column that we're using, so it could be gel filtration, it could be ion exchange, whatever, right? Absorbance at 220, most proteins absorb ultraviolet light at 220 nanometers, so we can measure how much protein is coming out the bottom here, right? As the pump is pushing everything through, and we get these very well-defined peaks here at 5 minutes, here at 7 minutes, here at 7.5 minutes, right? Each of these peaks is when a protein came through. Every time that this dips, that means that no protein came through, right? So here in this particular fraction, right, we know there's protein here, and this is peak here, this fraction, we have a fraction here, so we get high resolution with this high performance liquid chromatography. It allows us to use these finer beads. Yes?
Why is it in 220? Is it measuring light?
Yes, measuring light. So basically, most proteins absorb ultraviolet light at 220 nanometers, right? So we're using an ultraviolet spectrometer. So basically, we have a light source over here, right? A little light bulb. And we can tune it to emit light specifically at 220 nanometers, and we know exactly how much light that light bulb is putting out. So by putting a detector on this side, right, well now it takes how much light it sees minus, well it's the other way around, how much light it produced minus how much light it sees, that's how much light must be being absorbed by whatever's in the way. And what's in the way is what's flowing through the column, right? So as flows through, well now we get high absorbance, right? So that means that there's some protein here absorbing, in this case 0.2 or 20% of the light, let's say, right? All right, now other analytical techniques, we have protein electrophoresis, specifically SDS page. Okay, this allows us to actually visualize our protein. So all we've done so far is to purify our protein, okay, and do assays on it. But if we want to visualize and see, exactly how big it is and other types of properties, we need to be able to separate it. Now, in the case of SDS page, right, this is an analytical technique, whereas chromatography was a preparative technique for isolating the protein to use later on. So what is SDS page? Well, this relies very much on something that a lot of you have probably done, the same principle as DNA gel electrophoresis, only now we're doing it with proteins. Instead of agarose, what we're going to be using is something called polyacrylamide. Polyacrylamide is going to form the base for this gel. And the principle is the same, okay? I keep saying the same, but I haven't told you about it. I'm assuming you know. Has everyone heard of DNA gel electrophoresis? Yeah. Okay, good, all right. So we have this gel that's formed that is porous, right? At the top here in the wells, we put our protein sample, which might contain many proteins, could have 2, 3, a dozen, 100 proteins, okay? And we apply an electric field, right, with negative on the top and positive on the bottom. So everything that is rather negative, right, will migrate towards the bottom, the positive electrode. And that's what we see here. And because it has to wind its way through the gel, right, which is made up of all these tiny strands of polyacrylamide, the smaller proteins in this case are able to more easily make it through all of these, whereas the larger ones, because they have more mass and longer length, they have to kind of twist themselves around these polyacrylamide fibers, okay? So it takes them longer to migrate the same distance, so we get separation on size, right? So don't get confused between this and gel filtration. Gel filtration, the faster ones are able to move more quickly. In this case, smaller ones are able to move more quickly because it's not a going around situation, it's a going through situation. In this case, they all have to go through. Whereas in gel filtration, the big ones didn't have to go through. But now because they do, it's going to take them longer, right? So now we get separation based on size. The smaller ones are towards the bottom, this positive electrode, while the bigger ones are at the top, towards the negative electrode. But what's the problem with doing this, right? There's one big problem with doing this. This is always going to result in the same outcome. with all of our proteins moving down. You don't know if they're moving up. Well, they will always move based on size, but you're on the right track with the charge, right?
Repeat the question.
Is our protein sample that we go to the top, right? Is it always going to move down? Wow. Right. Proteins are not necessarily negatively charged, right? For DNA, right? DNA is always negatively charged as a rule. But proteins, remember, they can be neutral, they can be positive, they can be negative. So if we have all negative proteins, then fine, right? We don't have to worry about it. Okay? If they're positively charged, well, they're going to move up and out, right? So they're not going to enter the gel. Additionally, the protein is not necessarily always, among negatively charged proteins, they're not always evenly negatively charged. So we need a way to standardize both that proteins are negatively charged and that they're uniformly negatively charged, right? And we do that with this, SDS, sodium dodecyl sulfate. Sodium dodecyl sulfate is literally just soap, right? The FDA name for it is lauryl sulfate or sodium lauryl sulfate. They find it in shampoos and soaps and all over the place. I guarantee if you look in your shower, you're going to see lauryl sulfate. This is just SDS. And what SDS does is very interesting. Okay, it does two things, two very important things. First, it's a detergent, it's a surfactant. Okay, and what does a surfactant do for a detergent? Lowers the surface tension, okay, by doing what? Now, why do we have to use soap when we wash our hands? What does a detergent do? Huh?
Get rid of stuff.
Get rid of stuff, sure. Right, what kind of stuff specifically? It can break down membranes, all right, but there's one chemical principle that allows all of these things to happen, right?
It's polar and non-polar. So
Right, it's polar. Exactly, it's polar and non-polar. So it can help to kind of solubilize, as we say, forms an emulsion between non-polar molecules and an aqueous solvent, right? So it forms these micelles. We'll see that when we talk about membranes. So this is why we use soap for our hands. So what is it doing? It's essentially negating the hydrophobic effect, right? It's negating the hydrophobic effect. The non-polar tail sticks to non-polar molecules, and the polar sulfate here interacts, forms a hydrogen bond with the water, okay? Well, let's think about proteins. What do proteins, or what drives protein folding maybe? It's the hydrophobic effect. So by adding a detergent, negating the hydrophobic effect, what happens? We destroy the tertiary, well, the quaternary, tertiary, and secondary structure of a protein, right? So instead of a globular protein, now we have a polypeptide that is just long, right? Now we know that it's going to run based on size, because we can imagine a situation where we have two polypeptides or two proteins, both the same length, but one was completely alpha helical and the other was completely globular, right? The completely globular one has less surface area, so it would be able to move faster, whereas the alpha helical one would be stuck at the top, right? Now by adding SDS, we make them both the same size, so they should run the same. Additionally, look at the dodecyl sulfate part of SDS. It's got a negative charge, right? So it will negate the hydrophobic effect, but it also coats the protein and imparts a negative charge, a uniform negative charge across the entire length of the protein. So now we have fully denatured protein, and we have uniformly negatively charged protein. So we're guaranteed that if we apply a positive charge at the bottom and a negative charge at the top, we will always get the situation with proteins moving from top to bottom and separating based on the length of the polypeptide chain, right? So we can see an example of that here. Here's proteins separated by SDS page, right? Now, what are we seeing here? So we have our ladder, This is just something that you buy, okay? It's just a series of proteins for which the sizes are known, right? So that we can compare where is our protein in the gel versus where is a similarly sized protein in the ladder, and we can estimate the size of our protein that way, right? But these proteins are blue, right? Proteins are obviously not blue, right? So when we run our gel, right, we're just kind of hoping for the best here, right? Because we can't see the proteins. Proteins in solution are colorless. So what we have to do is to stain our gel, right? Stain, the most common one here being Kumazi. Kumazi stains specifically proteins, right? This stain binds to proteins and not to the gel. So when we add our Kumazi stain, now we see exactly where our proteins are. And we can compare the sizes of our proteins to the ladder. So for example, here this protein is running a little bit higher than 50, so we say it's maybe 60 kilodaltons. Here, right, we have to come over here and say, all right, that's about 30 kilodaltons and so on. So it's important both to have the ladder after FTS page, or during FTS page, and to stain the gel afterwards so we can actually see the results of our electrophoresis. Now, we can use an analytical technique like SDS page in conjunction with a preparative technique like chromatography or protein purification. We can use an analytical technique to monitor how our purifications are going or how they went, right? So say that we start with our homogenate, okay? So we have all these, tons of proteins. That would look, that would be something like over here, right? We have all these tons of proteins just from our homogenate, and then we do our fractionation, right? So our fractionation, now we've cut down on some, right? We have only cut down though based on, what cellular compartment those proteins are found in. So we'll still have the full range of sizes and charges and whatever, okay? So notice we have fewer proteins over here. Now, do our first chromatography step, ion exchange in this case, right? So we'll do our ion exchange chromatography. So what's happened? Well, again, now, we've taken all from our, say, mitochondrial fraction, and we've only isolated, let's say, the negative ones. We still haven't done anything with size, so we still have the full range of size, okay, but we just have fewer proteins because we've gotten rid of about approximately 2/3 of them, well, maybe a half, it depends, right? So we have fewer proteins here because we have purified them, but we haven't touched the size yet. The next step here, gel filtration, notice what's happened, right? We've gotten rid of everything bigger than here, and everything smaller than here, but everything else is the same, right? Still have a band here, still have a band here, here, here. This one was too little, so we lost it. Too little in amounts, not in size. Here and here, right? So we've kind of just cropped everything else. We've gotten rid of everything way bigger and way smaller. And then lastly, our affinity chromatography, right? So we take our gel filtration fraction, do an affinity chromatography step, and now we've gotten rid of everything that doesn't bind. It has nothing to do with size, but this is just due to the affinity of whatever protein we're working on to the substrate. Now we wouldn't want to use this, right? In fact, we can't use this because it's been denatured, okay? But we still should have some of our fraction left from our affinity chromatography, but this is just confirmation that we have purified successfully our protein. So whatever sample this lane came from should have only the protein that we're looking for, right? So we can use both types of methods in conjunction, analytical and preparative. Now, isoelectric focusing is an interesting type of analytical technique because it combines a couple of different topics that we've talked about, and it allows us to analyze many hundreds of proteins at the same time, which is not the case with STS-PAGE. So here, for example, in this homogenate, we have, just tons of proteins, so we have hundreds of proteins here. We can't isolate very many bands, right? So here, for example, we have just so many proteins running close together that's a smear over here as well, down here, right? Isoelectric focusing and two-dimensional electrophoresis allows us to measure all these hundreds of different proteins at the same time. So let's look first at isoelectric focusing. Who remembers what the isoelectric point of something is? We've talked about isoelectric point. Have we talked about isoelectric point? No. No. Okay. Good. So let's say that we have a tripeptide. I'm not going to to draw the whole structure in the interest of time. But let's say that we have a tripeptide made up of alanine, lysine, and histidine, okay? Of course, going from N to C. So we'll start our N terminus, go over here, and then I'm gonna draw it very badly. I'm not gonna draw the actual peptide bond or anything like that because they don't matter for this discussion because they're not ionizable. So we have our first C alpha from alanine, we have our second C alpha from lysine, and we have our third C alpha from histidine, and then here we have our C terminus. Alright, so again, I haven't drawn this correctly. I've ignored the peptide bond just in the interest of saving space here, right? So alanine is just a methyl group. Lysine ends in an amino. Histidine has the imidazole ring, which can be protonated here. And this, of course, the terminal carboxyl and the terminal amino can be protonated. So, the isoelectric point is related to the fact that the pH of any weak acid, or rather the charge of any weak acid, will change based on the pH. All right? So if we start at low pH, let's say pH 0, right? Well, that's high concentration of protons. So any ionizable groups that can be protonated will be protonated. So which are our ionizable groups? Again, we have this amino down here, we have this, and the carboxyl. Okay, so if everything is protonated, we have NH3+, NH3+, NH+, and COOH. We have a total charge of +3. Okay, now if we begin to increase the pH, right, we'll start to hit a pKa value. So what are the pKa values here? Let's say that for H3N+, the pKa value is 8.0. Over here is 10.4 for the lysine, is 6.0 for the histidine proton, and 3.2 for the carboxyl proton. Right? So the first pKa that we encounter is 3.2. And remember, what's going to happen now if we raise the pH above the pKa? What's going to happen to that proton on the carboxyl? It's the same as any weak acid problem. It's going to be deprotonated, right? So we'll lose this proton, and this becomes COO minus. So now our charge is +2, right? We keep increasing, we hit the next pKa value, which is 6. So we lose this proton, and this becomes just N, so we have a +1 charge. We hit the next pKa value, which is 8.0. And now we lose this proton. So we have H2N, we have a 0 charge, and we keep going here, and we hit the final pKa at 10.4, so we lose this proton. This is NH2, and now we have a -1 charge, right? This happens after we have pH 10.4. So now we can see that at any given pH, we have a different charge on this structure. We can have, say, a pH 4, a +2 charge. At pH 7, we have a +1 charge. But at what pH exactly will we have a 0 charge? This is the isoelectric point, right? So what we need to do is look at where we have the 0 charge structure and take the average or the midpoint between the two pKa values. So in this case, our isoelectric point, Pi, is simply 10.4 plus 8/2, or 9.2. So at pH 9.2, all right, 99.99% of our tripeptide here will be in the 0 charge state, okay? So let's go back to isoelectric focusing, right? We've set up a gel, okay, a tiny little gel, that's got a pH gradient, right? So we have low pH on the left and high pH on the right, okay? So what's gonna happen now? Let's add our sample either to the left, to the right, or in the middle, doesn't really matter, right? So let's say that we add it to the right, okay? Well, now, everything should be positively charged, okay? At least, everything should be positively charged. So what's gonna happen? Well, if we apply a positive electrode, positive electric field on this side, well now everything that's positively charged will be pushed that way. And if we put a negative charge here, everything that's positively charged will be attracted this way, right? But as it's moving this way, it's also moving up in pH. So the charge is going from positive to down to 0, right? So if we put our tripeptide over there that we did, right? So on pH 0, it's got a +3 charge, and we apply a positive and negative field respectively. Well, now it starts moving over here until the pH reaches 9.2. At pH 9.2, now the charge is 0. So of course it won't be repelled by this or attracted by this, so it'll get stuck there, okay? If we had it over here, the same thing applies. Now it'll be negatively charged, so it'll be repelled by this and attracted by this, and it'll move this way until it reaches 9.2, where it's got a 0 net charge and it stays there. So no matter where it is, it focuses at its isoelectric point, right? This is why it's called isoelectric focusing. Now, we get this arrangement here, so we can apply, you know, hundreds or thousands of proteins, whatever, and they will all focus at their isoelectric point. But the only thing that's the same between all of these is that they have the same isoelectric point. So we might have a protein that's 100 residues long with the same isoelectric point as a protein with 1,000 residues, right? So we don't know anything about the size. All we know is that all of these proteins here have the same PI, same over here, same here, same here. We can take this isoelectric focusing gel and now stack it on top of an SDS page gel, right? So we have our two-dimensional situation here, right? Our first dimension was IEF. Our second dimension is now isoelectric focusing. We apply a negative up here, or rather a positive and negative down here. Other way around, sorry. A positive down here, negative up here. So now, just like an SDS page, everything will migrate down. But now all of these proteins with the same PI will separate based on size as well, right? So we get all these little spots, and the same over here, and the same over here. So now we have our two-dimensional gel, right? And let's look at what a two-dimensional gel actually looks like. It looks something like this, right? Here, our first dimension was isoelectric focusing, and then we ran this way SDS page. So we can see all these spots separated by both size and isoelectric point. And this you can see we get much higher resolution, right? We can see many hundreds of different proteins here. In fact, you can use this to actually compare between different samples or different states, right? So we can look at, you know, which proteins are maybe expressed more highly in tumor tissues, or which ones are turned off in cancerous cells, right? So it's an analytical technique that allows us to compare different types of samples, but with high resolution, okay? Right, now we can quantify protein purification as well, using these units over here: total activity, specific activity, yield, and purification level. Total protein is easy, right? So let's go through different purification steps. Total protein is just how much protein we have. This is easy to measure. Again, the absorbance, say, 220 nanometers. We can say, all right, we're starting with 15 grams of protein total, right? So we have a total mixture in our homogeneous of 15 grams. Total activity is given in units, right? This is kind of funny, right? So it just puts units. That's because the units depend on the assay that we're using to measure how much of our protein we have, right? So different proteins will have different, you know, units to determine activity. So in our case, total activity in units, we have 150,000 units of activity given whatever assay that we dreamt up, right? Now, specific activity is equal to the total activity divided by total protein, right? So essentially what this is telling us is how much of our protein we have compared to how much protein in total, right? So in this case, let's just call it 10. So our specific activity is 10. Our yield is total activity divided by our initial total activity. In other words, how much of our protein do we have at that step compared to the amount of protein that we started with, or our protein that we started with. So at the beginning, it's always going to be 100%, right? We have 100% of the protein that we started with at the beginning. And the purification level is equal to the specific activity divided by the specific activity initial. In other words, right, how much of our protein do we have per volume, in effect, right? So how much more pure is our protein compared to everything else, right? So let's go through just a couple of these steps, right? So in fractionation here, right, we've lost, a good amount, about 2/3 of our protein. We're down to 4.6 grams. Total activity has dropped to 138,000 grams. So how much of our protein do we have compared to other proteins? Well, now our specific activity has gone from 10 to 30, right? So we have more of our protein compared to other proteins. So we have increased how present, right, as a ratio, our protein is. But how much did we lose? Well, we started with 150,000 units of our protein's activity, but now we only have 138,000. So 138 over 150 gets to 92%. Purification level, right, 30 divided by 10, is telling us that we have, as a ratio of our protein to everything else, we have 3 times more of our protein compared to everything else. Right? The same goes on, right? So down here, okay, we only have 68 milligrams of protein left, but we still have 75,000 units, right? Which means that our specific activity is what? Total activity divided by total activity divided by total protein, so we have 1,100, right? So we have 1,100 times essentially more, right? Or 110 times more than we had at the beginning. But our yield has gone down, right? 75 over 150 is 50%. Okay, so our yield is telling us how much protein we lost. Our specific activity is telling us how much of our protein we have compared to total amount of protein, and the purification level is telling us how many times more pure is our protein. Notice the dramatic step, by the way, going from gel filtration to affinity chromatography. Here we had a purification level of 110, and affinity chromatography, we had a purification level of 3,000. Okay, affinity chromatography is extremely useful in purifying proteins. In fact, sometimes that's the only step that we do. Here's one that we did in the lab, right? So we started with homogenate here, okay, just hundreds of thousands of proteins. We only did one step of affinity purification using an antibody, and we got this level of purification here. So affinity purification is extremely useful in doing quick purification. Now, if we wanted to do a really fine purification, we could have taken this affinity purification fraction and done then gel filtration and ion exchange and things like that. But this was good enough for what we need. Very often, affinity chromatography is good enough for what most people need. All right. Velocity centrifugation is another way to categorize not just proteins, but in fact many molecules. In fact, bigger molecules are typically characterized using what's called Ch.4 units, right? So sedimentation coefficient quantify the rate of movement when exposed to a centrifugal force, right? What does that mean? Right? So we take 2 solutions. One is high density, one is low density. We can mix them together in such a way that we can drip it into a test tube so that the bottom is of higher density and the top is of lower density. So we have this density gradient. We can layer our sample, protein sample or macromolecule sample, doesn't matter. We can layer that on top. And what's going to happen here is we'll spin it at high speed. And the speed at which this moves through this density gradient is what determines what the Ch.4 unit of that particular molecule is. Okay? So, dense molecules, or not dense, but large molecules, let's say, ones that move slowly through here have the larger Ch.4 units. So the less it moves through the density gradient, the larger the S value. The more quickly it moves through this gradient, the smaller the S value. Okay? So you might have heard, the 30S ribosome and the 50S ribosomal subunit, right? The 30S and the 50S. They come together and they make what? The 70S. Right? So Ch.4 units are not additive. Right? So how do we figure out these Ch.4 units? Well, we took individual, small ribosomal subunit. We did velocity centrifugation, and we discovered that it had a Ch.4 unit of 30, and we did the same for the large one. Then we did the same for the complex. Right? So we saw that we had a 30S, a 50S, and a 70S complex. They're not additive because shape is a factor. Right? Same as what we saw why we had to use SDS in SDS page. If we have 2 proteins that are the same length, right? So they have the same mass, but one is spherical and one is alpha helical. The spherical one will move more quickly through, so it will have a smaller Svedberg value, whereas the helical one will move more slowly, it will have a higher Svedberg value, right? So the Svedberg values tell you not only about the size of a molecule, but also the shape of the molecule, right? So they don't add. When we add the 30s ribosomal subunit and the 50s ribosomal subunit, now it's got a new shape, which has to be taken into account. And that shape just makes it as a Group 7S. All right, antibodies, again, are powerful tools to study proteins. We've seen how antibodies can be used in affinity chromatography, but antibodies can also be used in conjunction with other techniques, in this case, an analytical technique, SDS page, something called a Western blot. So we look at an antibody, all right, it just looks like this. We don't have to get too bogged down with the structure of an antibody, but essentially it's got two antigen binding sites at the ends here. It's made up of two chains, it's made up of four chains rather, two heavy chains here in blue and two light chains here in red. They're held together by disulfide bonds, and they have antigen binding sites at the ends here of this Y structure. Now, why do we care about antibodies? Right, so let's skip all the monoclonal and polyclonal. Let's skip these, right? Because we can use antibodies quite effectively in many different types of analytical techniques, specifically in ELISAs. So ELISAs are very important in the lab somewhat, but even more so in the medical field, right? What type of tests rely on ELISAs? Does anyone know? Has anyone heard of any test that relies on this method called ELISA?
Cytokine levels. Cytokine levels.
That's probably one of them, right? But there's a couple of very well-known ones. HIV, sure, right? And how does an ELISA work, right? Well, we can detect using either an indirect ELISA or a sandwich ELISA. They're used for different things, okay? An indirect ELISA is used to test for the presence of an antibody, right? This is very rare, right? We don't typically see this, but essentially we coat the bottom of our test chamber with an antigen. We add our sample, which we're testing for an antibody, so the antigen should bind to the antibody. Then we get rid of everything else, we wash it. We add a secondary antibody, which is going to recognize the primary antibody, and this secondary antibody has some reporter enzyme on it, right? It could be something that changes color or that catalyzes some reaction. In this case, it catalyzes a reaction that turns the solvent yellow, right? So if we have our secondary antibody binding, we should have the reporter enzyme, so we should get the production of this yellow color. If we didn't get any binding of the primary antibody, right? Well, then when we added our secondary antibody, it wouldn't bind to anything, and after we washed it to get rid of everything, we wouldn't have any reporter enzyme left over, right? So we wouldn't get the change in color. By far the most common type of ELISA, though, is this sandwich ELISA, okay? And what we do is we coat the bottom of our reaction chamber not with the antigen, but with an antibody that recognizes an antigen that we're testing for, right? What happens? Now we add our sample, okay? This contains our antigen, let's say. It either does or does not contain our antigen. This antigen is going to bind to our antibody, which is already there. Now we'll wash to get rid of everything else. So if the antigen was there, it's bound. If the antigen was not present, then there's nothing left over here. We add our secondary antibody, and our secondary antibody also binds the antigen, but at a different site than the primary antigen, or antibody rather, right? So we have this antibody binding on this side of the antigen, and we have our secondary antibody binding on the top of the antigen, right? So now again we wash. So if this bound after the wash, this will still be stuck, and we'll get our report here. And this is the type of ELISA that's used in pregnancy tests, testing for HCG, in COVID tests, testing for, I don't know what they're testing for now, usually the spike protein, right? But, you know, we don't have this situation here where we have like a test tube and we have to move solutions over. Instead, they've devised this kind of very clever system here where we have a capillary pad or a wicking pad, right? And we apply a sample to one end, and just through capillary flow, right, it moves through all of the stages that we just talked about, right? So here we have our antibodies conjugated to a tag, right? So it's either gold or latex or something that fluoresces, whatever. And as this sample now moves through them, it will take all these antibodies with it, right? And all these antibodies, if they are binding, right? If they're binding to the antigen, well, what's going to happen here? Now we're going to have this situation where we have the antibody bound to the antigen, right? Of course, we have excess antibody, which is fine. As these move, the first thing they're going to hit is the test line, right? They hit the test line, and what happens? Now, this binds the other side of the antigen, right? So it'll hold it there. And now this reporter enzyme, the yellow, is on the test line, and that will turn some chemical blue, right? So that's what you see when the line turns blue in a COVID test or in a pregnancy test. It's the presence of all these reporter enzymes here. If this didn't bind the antigen, so there was no HCG or no spike protein, well, now as this kept going, it wouldn't bind over here, so we wouldn't have any of these, so the line wouldn't turn blue, right? Now, as this continues on, it'll also hit the control line. The control line is not testing for the presence of the antibody, it's testing for the presence of the primary antibody. So this should turn blue whether or not there was antigen present. The reason that we want the control line is we want to make sure that there was functional antibody at the beginning here, right? If the control line doesn't test blue, right, that means that no antibody was detected, so it doesn't matter what happened over here. Nothing can be, nothing can be said about it, right? So this is, again, by far the most common way that ELISAs are done, right? This is how ELISAs are run, just on these wiki pads. We can also use antibodies for Western blots, right? So Western blots are just a conjugation of SDS page, SDS page, that's it, SDS page. and using antibody, right? So we run our sample. Now, we could use, again, SDS page to look, to monitor our stages of purification, but say that we don't want to be fussed with that. And all we want to see is, is our antibody there and how much of it do we have? Well, if we just ran a homogenate, okay, well, we would just get a big smear of proteins as we've seen before, so that wouldn't be very useful. So how do we pick out from that smear our protein? Well, when we had something that binds only our protein, that could be an antibody. So after we've run our SDS page, right, let's just look at one lane of the SDS page, okay? We'll transfer this onto a polymer sheet. This is something like nitrocellulose or PDDF, right? What this polymer sheet does is it binds to proteins specifically, okay? So when we transfer the proteins over, right, we just stack the nitrocellulose sheet under the polyacrylamide gel, and we just transfer everything down, right, through the gel onto the nitrocellulose sheet, so we get exactly the same patterns of proteins as what we started with. To this now we add our antibody. Our antibody is going to recognize our protein, right? But this is still not done because we have no way of visualizing either the protein or the antibody. Even if we tried to stain this now with Kumasi, it would still show all of these proteins and our antibody. So staining is out of the question here. Instead, what we do is add a secondary antibody, similar to what we saw in ELISA. This secondary antibody is going to recognize our primary antibody. And this secondary antibody also has a reporter enzyme, usually something called horseradish peroxidase, which produces X-rays, right? X-rays can be detected on an X-ray film. You can get the X-ray of the doctor, and now the only place that we should have X-rays being produced or exposure on our film is where our protein is. So we can go from a situation where we have potentially hundreds or thousands of proteins here to a situation where we have only one band because that's where X-rays are being produced. You can also use antibodies for in situ hybridization, right? The reporter enzyme doesn't have to be something that produces X-rays or something that produces a color. It's something that can produce light as well, okay? Here, we're using 3 different antibodies specific to 3 different proteins that have reporter enzymes that are producing 3 different wavelengths of light, green, red, and blue, right? So in the blue here, right, this is likely some antibody that recognizes a nuclear protein, okay? And that nuclear protein, or that antibody was conjugated to a reporter enzyme that gives off blue light. And the same could be said here for actin, or let's call these probably ribosomes, right? So we have these in situ hybridization. You can visualize where in the cell proteins are, right? For small animals, like flatworms and C. elegans, you can actually do this in a whole organism, right? In a whole multicellular organism, right? You can use these fluorescent antibodies to do in situ hybridization and see where your protein is. You can use protein or antibodies as well to do what we call co-immunoprecipitation. Co-immunoprecipitation allows us to discover what partners, if any, a protein has, right? So is our protein interacting with any other protein? Well, let's say that we want to figure out, right, which partners this red protein has. We don't know. We do have an antibody for the red protein, but we don't know if that red protein is binding to any other proteins and the identity of those proteins. Well, if we take our antibody, right, we add it to our solution here, which contains a bunch of proteins, including our protein plus potential binding partners. Well, now we'll get binding to the red protein, right? We can add it's a special type of resin called protein A resin, right? These just bind non-specifically to antibodies, right? So we add our antibody, which binds to our red protein. We add our protein A resin, which binds to those antibodies. And because this resin is a resin, now we can just centrifuge it. That will pellet the resin all the way down at the bottom, okay? Everything else remains in the supernatant. We can get rid of the supernatant, isolate this pellet, and now we can identify what proteins are present. If a protein is present in the pellet down here, that means that it must have been binding to our red protein, and we can identify partners that way. But how can we actually identify those proteins? Well, this is done using something called mass spectroscopy. Mass spectroscopy can be used to identify proteins. So let's examine how this looks. In context of proteins, right? I know mass spectroscopy is its own field, but I'll just try to go over this briefly. In the context of protein chemistry, we use this MolecBio type of spectroscopy. MolecBio standing for matrix assisted laser desorption and ionization. And essentially, the analyte, in our case, the protein mixture, is vaporized by a laser beam in this matrix, right? So this matrix here, which is supporting our protein mixture, is gone, it sublimates, and what's left are ionized proteins, right? So they have a charge. These ionized proteins can then be fed through a mass spectrometer, and the amount of time that it takes for those ionized proteins to make it through this mass spectrometer depends on both their mass and their charge. And what we measure is, well, we know exactly when we vaporized that protein, and so we know exactly when it entered this tube here, right? It's got an electrical field. So by measuring how long it takes to hit the other side at this detector, we can effectively measure the mass to charge ratio, right? So how much mass versus how much charge. And get us to plot something like this. So we have a mixture of, say, insulin and beta-lactoglobulin, right? Well, what do we see? Well, we see all this here, and then eventually we get a jump here, intensity, at 5733.9, right? So this is insulin in the 1+ state. If we multiply that by 2, we get insulin in the 2+ state, right? So at 5733, instead here we get at whatever that is, times 2, right? What does this mean? Well, it means again that we're only measuring the mass to charge ratio, but it allows us to separate different protein populations within a mixture of proteins. So if we come back here to our co-IP, we can now take this pellet, we can separate out all of the proteins, right? Just by adding a little bit of STS or what have you, we can then subject this mixture of proteins, which in this case contains only red and yellow, we can subject that to mass spectroscopy, and we get 2 peaks, right? We would get 2 peaks because it would have different mass to charge ratios. But again, this only tells you the mass to charge ratio. It doesn't tell you the identity of those proteins. For the identity of those proteins, what we need is to sequence those proteins. And we do that using a method called tandem mass spectroscopy. These are in the wrong order, so I'll come back to this one. In tandem mass spectroscopy, right, we do our first step here, right? So again, we take our peptides, and we put it into the mass spectrometer, which goes through that MolecBio situation, ionizes them, and then we get our mass to charge. So each one of these peaks should correspond to a peptide, right? It should correspond to a protein. Each one of these peaks I will talk about on Tuesday, because everyone's dying to get out of here, it's 11:39 on Friday. So we'll pick back up with tandem mass spec on Tuesday, right? 'Cause I do want to get through it, it's very interesting.